Hi, good evening. I'm here with another video. So, in this video, I will be discussing KCET 2000, KCET 2023 paper, 2023 biology paper. So, directly we will start with the questions without wasting any time. So, read out the first question. Read out the first question here. What is the function of protein GLUT4? So they are asking regarding the function of proteins. So this is from a biomolecule chapter, that is first PU syllabus. So in that chapter they have given a protein functions in a tabular column. This GLUT4, it is one of the protein. It facilitates or helps in the enabling the glucose transport. This enables glucose transport into the cell, transport into cells. So let's see what are all the options they have given in this question. In a, act as an enzyme. No, the enzyme, that is trypsin, will act as enzyme. That is one of the function of protein, so this is wrong. Enables glucose transport into cell, so this is true regarding this one. Sorry. Enables glucose transport into cells. So next one, fights infectious agents. So this is antibody. So GLUT4 function is enabling the glucose transport. Here fight infectious agent, that will be, so I will write here. So act as enzyme, that is a trypsin will act as enzyme. Here fights infectious agent, that is antibody. Then T functions as intercellular ground substance. So this is also not a function of a GLUT4. So B is the right answer for this question. So 1, B is the right answer. Let's discuss the next question, that is cells in the quiescent stage, that is Z0. So this is a cell cycle. Consider it as a cell cycle. So where the cell cycle, you will find two phases. One is interphase, another one is M phase. Interphase, you will get again three divisions, that is G1 phase, S phase, G2 phase. Then in a M phase, you will again come across four different PMAT stages: prophase, metaphase, anaphase, and telophase. Then here it will be called as C0 or the quiescent stage. So here in interphase stage of the cell cycle, this one G1 to G2 phase, this will be interphase. Here the formation of different things in a cell takes place, like protein synthesis, DNA replication, macromolecular formation, etc. It will take place in interphase. When the cell enter into this M phase, the cell undergoes cell division. Soon after this, in telophase, the cell will be divided into a cell will get divided at the end of the telophase. Soon after this, these cells may enter into the Z0 or they may enter into the cell cycle. So, if the divided cell enter into Z0, then this Z0 or the quiescent stage is also called as a resting stage, where the cells, they remain metabolically inactive. They will not do any process, any activity if they enter into the quiescent stage or the Z0 stage. Hence C is the right answer for this question. That is, they remain metabolically inactive. Let's discuss the third one. Consider the following statements 1, 2, and 3 regarding criteria for essentiality of the nutrients in plants. So, this question is basically from mineral nutrition, but that chapter has been removed from the syllabus. So, consider it as a out of syllabus. So, we'll directly discuss the fourth question. During chemiosmotic synthesis of ATP, during chemiosmotic synthesis of ATP in photosynthesis. So, what actually happens in this chemiosmotic synthesis of ATP? So, they have given four different options. Let's discuss which is appropriately explaining regarding the chemiosmotic synthesis. A. The protons accumulate in the intermembrane space of the chloroplast, so this is wrong. So the protons will not accumulate in this intermembrane space. Then B, the proton gradient is not required. So proton gradient is actually needed in this chemiosmotic hypothesis. Or the So this one is also wrong. C, the protons accumulate in the intermembrane space of mitochondrion. So this is wrong. So this chemiosmotic synthesis takes place in chloroplast. They have given mitochondria. So this one is also wrong. Then D, the protons accumulate within the lumen of thylakoids. So this one is true regarding the concept where the protons they will get accumulated in the lumen of the thylakoids. So D is the right answer for this question. Next question, fifth one. Fifth question. When a tripalmitin is used as respiratory substrate in aerobic respiration, the process consumes 145 molecules of oxygen and releases 1 or 2 molecules of CO2. Then RQ value could be. So they are asking regarding the respiratory quotient, that is RQ, that is a respiratory quotient. So RQ equals to, we have a formula that a respiratory quotient equals to a volume of CO2 evolved divided by a volume of O2 consumed. So I will write clearly once again. CO2 evolved to the CO2 consumed. So in this question they have given a volume. So RQ equals to volume of CO2 evolved, that is 1 or 2 molecules of CO2, 1 or 2 molecules of CO2 evolved divided by 145 molecule of O2 consumed. So RQ value will be what? Calculate this one. So you will obtain 0.7. So this RQ value of this tripalmitin, tripalmitin, that is a fatty acid. So this fatty acid RQ, that is respiratory quotient, that will be 0.7. So B is the right answer for this question. So next question, let's discuss next question. So sixth question. Identify the incorrect statement with reference to biocontrol agents. So these biocontrol agents, they will be helpful to the nature and also to the crop field. They does not have any negative impact on the crop plants and also to the environment. So let's discuss this one. They have given four different statements regarding biocontrol agents. All you need to do is you have to choose this incorrect statement. So A, read out the A thing. They do not show any negative impact on crop plants. So this is true. They are nature friendly, crop friendly. So A is true. They help to increase the use of synthetic pesticide. So this statement is wrong here. So this biocontrol agent, it will not encourage synthetic pesticides. So this is incorrect. C, they can significant in treating ecologically sensitive area. This is also true with the biocontrol agents. Then D, they do not affect non-target pests. So this is also true. So B is the incorrect statement regarding biocontrol agents. Next one. A farmer has applied chemical fertilizers in his crop field for many successive seasons. In the next season, the crop growth was poor as the soil lost its fertility. Suggest the suitable microorganism that replenish the fertility of soil in his field. So here they are talking regarding the continuous usage of chemical fertilizer result into uh, degrade in the soil fertility. So in order to uh, replenish the same soil fertility, what should be used? So here the nostoc. So here the nostoc. So this nostoc will help in the replenishing the soil fertility. So B is the right answer for this question. So let's discuss the eighth one. So, in cloning vectors, antibiotic resistant genes are helpful in. So, this antibiotic resistant genes they help in selection of recombinants recombinants from non recombinants or transformants from non transformants from non transformants. So, they have given four different statements here. Let's discuss one by one. A transfer of foreign gene to the host. So this is wrong. The B option, selection of recombinants. This is true with this antibiotic resistance gene present in a cloning vector. So B is right. C, making the host cell competent. So this is wrong. Cleavage of vector by Aryan. This is also not appropriate statement regarding antibiotic resistant genes. So B is the right answer for this question. So next question. A student while extracting DNA from aspergillus fungus required dash enzyme to break open the cell wall. So as we all know, all living organisms will be covered by membranes, plasma membrane and cell wall. In order to extract DNA which is situated in a nucleus, we require different enzyme to degrade the membranes. So here the cell wall will be degraded by cellulase, then pectinase, proteins which are present in a cell, that will be degraded by pectinase, where the fungal cell wall will be containing this chitinase. So this fungus cell wall is formed from chitin. So in order to remove this chitin, we require a special enzyme, that is chitinase. So this chitinase enzyme will degrade the chitin of the fungus. So D is the right answer for this question. Next question. Identify the DNA sequence which can be cut using EcoR1. So here EcoR1, it is one of the restriction enzyme. So this restriction enzyme will recognize a particular sequence, that is palindromic sequence, that is GAA-TTC. So this will read the DNA sequence from 5 prime to 3 prime. So in this 5 prime to 3 prime direction, if the DNA sequence contains this palindromic sequence, that is GAA-TTC, then this EcoR1 will cut specific DNA sequence. So in this one, just find out where this GAA-TTC palindromic sequence is present. In A, GCTT, so this is wrong with this EcoR1. Here ACGAATTC. So this is true 5 prime to 3 prime. So this one is true with this question. Then C, TACTT. So this is also wrong. Then here ACGAATTC they have given, but here they have given the 3 prime to 5 prime. So this is also wrong. So B is the right answer for this question. Next question, that is 11th question. So read out the next question. 11th, which of the following amino acid is coded by single codon? So these triplet codons have many different properties where a single amino acid may be coded by a single codon, or the amino acid might be coded by many different codons. So here they are asking regarding amino acid coded by a single codon. Here valine, it also has many codons. Degeneracy will be shown by valine, phenylalanine, tyrosine, but here tryptophan. So this tryptophan has only one codon, that is UGG. So this single triplet codon will code for a single amino acid, that is tryptophan. Apart from this, another amino acid, that is methionine. This amino acid is also being coded by a single triplet codon, that is AUG. So if they ask tryptophan or methionine, then you should choose it to have a single codon will be coded. So 11th D is the answer. Next question, that is the 12th one. In prokaryotes, the transcription of DNA is initiated with the help of. So, in case of this prokaryotes, the transcription will be initiated by, the transcription is initiated by a factor, that is a sigma factor. So, this sigma factor help in the initiation of transcription in case of what? In case of prokaryotes. Remember, in prokaryotes, this sigma factor will go and bind to a polymerase. So that is RNA polymerase. So sigma factor is the right answer for this question. Then read out the next question, that is 13th question. According to Human Genome Project, that is HGP, the total number of genes in human genome is estimated at 30,000. The number of genes present on Y chromosomes are. So this Human Genome Project, they have estimated there are around 30,000 genes will be present. So they are asking regarding this Y chromosome, how many genes will be seen in this Y chromosome. So they have given, that is 231 genes will be seen in Y chromosome. Then in first chromosome, where you will get around 2968 genes. So in this Y, they are asking Y chromosome, then 231 genes is the right answer. So 13 C is the right answer. Next question, that is 14th question. 14th, in a crime investigation, the investigating officer collects different biological samples from the crime spot for DNA fingerprinting analysis. Which of the following sample is not helpful in the analysis? So, in order to do a DNA fingerprinting, the samples like the sample has to be a bone will be used as a sample, hair will be used as sample, then a skin will be used as a sample, then a sperms will be used as a sample, and also apart from this one, a saliva will be used, then the last one, that is blood, will be used. So let's know. So sample, so these are all the samples, so we can use here. They have given skin shreds, it can be used erythrocyte. So this one is not used for DNA fingerprinting because erythrocyte means RBCs. These RBCs, they doesn't have any DNA, they lack DNA. So due to this aspect, the erythrocytes will not be used. Semen sample, it will be used. Hair follicle, it will also be used as a sample for DNA fingerprinting. So erythrocyte, it is the right answer for this question because they asked which sample is not helpful in analysis. So erythrocytes are not helpful in the analysis. So next question, one last question for this video. A mature mRNA consists of 900 bases without any stop codon in between. Calculate the number of amino acids coded by this mRNA during the translation. So they are asking, there are 900 bases. These 900 bases will code for how many amino acids? How many? That is the question. So let's discuss in detail. So as we all know, in a single codon, you will come across three bases. In a three bases, then a single codon have three bases. Then here there are 900 bases. 900 bases will be having how many codons? It will be having 300 codons. If there are 300 codons, then it will code for how many amino acid? 300 amino acids, because a single base or three bases like AAA, AUG, CCC, GGG. So these are the triplet codons. This three bases will code for one amino acid. This will code for one amino acid. This will code for second amino acid. This will code for third amino acid. Here there are 300 codons, and these 300 codons, if 300 codons, then 300 amino acid. So the right answer for this one will be, that is 300. So for 15th question, C is the right answer. 300 is the right answer for this question. So we'll discuss some more questions in the next video. Thank you for watching.